We're very pleased to welcome Dr. Mike O'Shannon. Mike is an investigator of the Howard Hughes Medical Research Institute and professor of neuroscience at Columbia University. He obtained his BA from Brown and his PhD from UC Berkeley, where he worked with Ralph Freeman. He also obtained an MD from Brown, meaning that unlike most of us, he's an actual doctor. He then conducted postdoctoral training with Bill Newsom at Stanford, and finally joined the Department of Physiology and Biophysics at the University of Washington, where he remained until 2012 before joining Columbia. Mike has made fundamental contributions to our understanding of decision-making by combining behavior, animal electrophysiology, and computational modeling. Beyond his stellar scientific career, he is also an avid jazz guitarist. So let me keep this short, and without further ado, Mike, take it away.
Thanks a lot, Robbie. Thank you, Bilal. Thank you all for inviting me. I'm sorry about the technical problems that we can't see each other. So I'm going to talk to you today about some new work that involves a collaboration with these people, Zheng Wu, who goes by Herbert, and a theorist, Ashok Litwan Kumar, and another experimentalist, Richard Axel. This is, I want to just give you some background, which is that in evolution, the emergence of cognitive capacities was accompanied by an expansion of the cortical mantle. I'm assuming you can all see my mouse. And most of it's devoted to what we call association areas, and many of the neurons in these areas, especially the parietal and prefrontal cortex, exhibit persistent neural activity. And that's often associated with working memory or knowledge states, what I'll call gnosis, with the G, or praxis, like planning. And decision-making, I think, kind of holds the key to cognition, or a key. It offers us some traction on the elements, what Plato called carving nature at its joints. And so I think that my basic approach to neuroscience is that the principles that are elucidated in the monkey by studying decision-making, a pretty complicated decision-making, but still simple compared to the things we're doing right now, that when those principles are writ large, they're likely to underlie sophisticated cognitive processes that go on in humans and ultimately give us some indication or some insight into what's responsible for disorders of cognition in humans. Now, to treat those kinds of disorders and make a difference in brains, I think we need to understand those principles at a much more refined level than we're capable of learning in the monkey. And so I've been devoting a portion of my lab now to the mouse in order to sort of break things down. And the talk today is I'm going to talk about a monkey-inspired project that led to some work in the mouse. So I'll give you some background about decision making, and I'll introduce a problem that has interested me for a long time, but I think is difficult to study in the monkey, which is the control of the circuit configuration. I'll explain what that is later. And then we'll study that in the mouse using a simple delayed match to sample. Now, for years and years, I've been studying this problem, like trying to understand the brain one dot at a time. And we asked monkeys to decide the net direction of these random dots, like essentially which way is the wind blowing them. And we control the difficulty by controlling the probability that a dot's moving in motion or displaced, from moment to moment, as opposed to randomly replaced with the noise. And the monkey answers by making an eye movement in most of our experiments, sometimes a reach. Summarizing over 20 years of work now, we understand the computations and to some extent the neurobiology behind this. The idea is, stated kind of from a computational perspective, is that information, noisy information from the world, in this case the random dots, gets transduced by retina, lateral geniculate, especially cortical areas that have direction-selective neurons in primates, into what I will call the momentary evidence, moment-by-moment fluctuations that might favor right versus left, as represented by this noisy distribution on its side where this direction means evidence for right and this for left. And the idea is that you can relate that to the moment-by-moment spike discharge from neurons in the extrastriate visual cortex, especially area MT or V5. And I know there are experts on that area in the audience right now. Now, the idea is that neurons with longer time constants in the association cortex, they represent, over a longer period of time, the accumulation as a function of time of that momentary evidence. this might be the path that such an accumulation of signal plus noise takes until it stops in this absorbing upper bound, which means terminate the decision at this point in time with a rightward choice. Had it reached this bound, the lower one, it would be a leftward choice. Now, immediately you can see that this idea, this computational idea, explains the trade-off between speed and accuracy, because if these bounds were closer to the starting point of the accumulation, then say this trial would have ended in a left choice, which would have been incorrect, but at the savings of time, on average anyway. Okay? So it turns out that this framework does really well in explaining the what I'll call evidence accumulation or accumulation to bounds, sometimes called drift diffusion with bounds. This framework explains the behavior of the monkeys and humans beautifully. So if I plot on this graph the reaction time of the monkey, that's the time that elapses from onset of the motion to the beginning of the monkey's eye movement response. If we plot that as a function of motion strength, here's signs so that the positives mean rightward, the negatives mean leftward. So this is easy rightward, easy leftward, and the difficult cases in the middle, which are most of the trials, are in the middle. And you can see that when the motion is weak, the monkey takes almost a second on average. When it's strong, he takes about 400 milliseconds on average, okay? So that's kind of cool. The nice thing about this framework is that it gives us simple mathematics to explain essentially the first passage times of this biased random walk or drift diffusion. And t, being the reaction time, is explained by this equation. And very briefly, it only has two important parameters. It has a bound, which I've already introduced, which kind of mediates the speed-accuracy trade-off. It has just a single constant k here, which multiplies the motion strength, c, and converts it into units of signal to noise. And then there's some non-decision time, the part of the reaction time that's not spent doing this, deliberating on the evidence, okay? Now, that's the equation that's fit to this data, and that's the fit. But the cool thing here is that this mechanism, a computational mechanism at this point, but in a moment I'll show you physiology, that a computational mechanism, it reconciles not just how long it takes to make the decision, but the probability that that decision will be the correct one, or a rightward choice in this case. And so that function, that function is shown here is a logistic function. So the probability of right is a logistic function, again, of this product now of these two free parameters. Now these two free parameters were used to fit these reaction times. And now we have a complete prediction just based on this fit. We make this prediction for how the choices should go, so that say these two motion strengths and these two, they should lead to perfect behavior, all left choices, all right choices. And the graded change in behavior should go at exactly this rate according to this equation. And this is where the data live. So it's a pretty good job making set of predicting, not fitting, but predicting these choice proportions, but just by measuring the reaction times. And it gives us some reasonable reassurance that this is the kind of mechanism that's at play in the brain. We didn't invent this idea. It came from classified work in World War II, both from Abraham Wald, who was doing munitions quality control in the States, and from Alan Turing, actually, as part of his decryption of the Enigma code. I can tell you some about that in the Q&A if you like. But of course, it was used by many psychologists, mathematical psychologists, and I've just listed a few of them here that have been champions of these kinds of models, Roger Radcliffe and Stephen Link in particular. Okay, so when I was a postdoc with Bill Newsom, we thought we would try to find the candidate neurons that would read out the information from the visual cortex. And so we thought, okay, they must receive information from direction-selected, that's what the DS stands for, from direction-selected neurons, maybe directly, maybe indirectly. And they have to project to the oculomotor system, directly or indirectly, because ultimately the monkey answers with his eyes. And they should exhibit long time constants, like they should have persistent activity, otherwise they'd be incapable of computing something like the integral, which we suspected was going on, the accumulation of evidence, mathematically and integral. And so over the years, Bill, I, other labs as well, have studied the dorsolateral prefrontal cortex, the frontal eye field. We've been in the medial intraparietal area now. But I'm going to show you my favorite area, the lateral intraparietal area, LIP. Now, neurons in LIP, which is on the lateral bank of the lateral, of course, of the intraparietal sulcus, which I'm outlining here, but roughly where the orange is. They were identified by Richard Anderson as the part of Brodmann's area 7 that projects to the frontal eye field and the superior colliculus. So you can consider these neurons part of sort of our knowledge system, which is what we think of the parietal lobe, but they are involved in what we do with information with our eyes, like making eye movements to it or directing attention. And these neurons have persistent activity that's spatially selective. Let me illustrate that by showing you a very simple working memory task where we're going to flash a spot roughly here, and we're going to ask the monkey to make an eye movement to its remembered location. He doesn't get to see that, Gray. That's for you to remember where that spot was. But let's listen to neurons. Hopefully you'll hear them as the monkey performs this very simple working memory task. I hope you heard that. And I can't see anyone, and I don't hear anyone, so I have no idea if I'm just talking to a wall. Can someone just say that they've heard this?
We heard it.
It sounded great.
Great. Okay, good. So these are the spikes. The red spot came on and off. Monkey told to make his eye movement. Monkey made his eye movement. That was the yellow streak you saw. Okay? That's persistent activity. And if you ask me and many others, this persistent activity holds the hallmark, holds the key to understanding cognition, because sort of all of our mental acts that we would call cognitive have a freedom from immediacy. That is, we don't have to act in the time frame of the immediate changes in the world, nor do we have to keep up with a body in real time. So those are the real-time demands, but between that, we want some flexibility. And I think in evolution, that's what led to the inchoate versions of what we recognize as cognition in us. Now, in LIP, not all areas, the association cortex is just, chock full of these kinds of neurons. But in LIP, these neurons are spatially selected. They may have other properties in other association areas. So if I flash the spot over here, okay, now there is no elevation activity. So that's the persistent activity that's spatially selected. And what we do in the lab is we contrive tasks. They are contrived and not natural. We contrive tasks to study decision making by setting up experiments where one of the targets, one in the response field, will represent at least the choice that the monkey thinks that these dots were moving to the right and makes that eye movement, as opposed to making this eye movement. And when we studied those neurons, I think we got kind of lucky. And they didn't just show us the choice. They gave us insight into the decision process leading to the choice. Because when we study them in the reaction time setting, we know the window of time that the monkey's making his decision on every trial. And here I'm showing you just a few motion strengths. And these solid curves are the trials where the monkey chose the target in the response field. And you can see they're rising. And these, the firing rate, I'm showing you firing rate, by the way, is a function of time. 0 is when the motion was turned on. Sorry, I should have said that. These dash curves are the trials where the monkey chose the target outside the response field. Okay, these are averages of about 40 or 50 neurons. So, but this is true on the single neuron level as well. And what you immediately appreciate is that these neurons do tell us what eye movement the monkey's going to make. The solid curves are above the dash curves. But they're also telling us something about the quality of the evidence that he used to make the decision, because they rise faster for the strong motion. That's the one I keep showing you in the examples, 50% coherence. But they also they rise at a lower rate, or even meander, you might say, for this dash curve when the motion is weak. I mean, the monkey might as well be guessing because he's rewarded at random when we put no average motion signal in, but of course there are fluctuations in the display, and the monkey makes his answer based on them, and you see that revealing itself gradually as a function of time during the decision. This red set of curves is just one of the intermediate motion strengths, and you can see it occupies an intermediate level of rise and decline. Okay, so we get the idea that these averages might conform to, might be a representation of the accumulation of evidence for the target in the response field and against the target out of the response field. There has to be an against part because the responses can decrease in response from their starting point here when the process begins to unfold. Okay, so I'm just writing that as an integral. Okay, now this is the reaction time task. So these curves, are clipped at the median reaction time, and they cut out any activity near the time of the saccade. But what you can see is if we align the same sets of trials to the time of the eye movement, the saccade, when the monkey makes his answer, you see that these responses have come together in a very stereotyped way before the saccade. And if we look at all the trials and we just break up all the trials that ended in this choice for the target and the response, and in the response field, and we break them up and we group them by reaction time. So this is now the fastest quantile, and the blue, the indigo, is the slowest quantile. Now you really get the idea that they reach a common level about 80 to 100 milliseconds before the saccade begins. So we think this is a sign of a termination threshold, that something else outside of LIP is effectively reading off the spike rate of LIP or any of those other areas of it, that do the same kind of thing that LIP is doing, but they'd be highly correlated with LIP because at the time of that readout, LIP is roughly at 62 or 64 spikes per second, plus or minus, okay? So that's the sign of that bound or threshold, okay? Now, I know you might notice that these responses don't come to a stereotype level, and that's an indication that in the brain, we don't have diffusion between an upper and lower bound, but we have a race between a mechanism that, say, has the rightward choice target in its response field, so it's accumulating evidence for right and against left. But there are other neurons, mostly on the other side of the brain, that accumulate evidence for left and against right because the left choice target is in its response field. And of course, right minus left and left minus right, at least at the level of the video monitor, are perfectly anti-correlated. They may not be perfectly anti-correlated in the brain, but we'd see some process like that. So when we record from a rightward preferring neuron when the choice is leftward, we see something like this coming from a rightward neuron, okay? Or vice versa. All right. Now I want to say this idea generalizes, and it probably is not so simple as integrating a difference in firing rates from MT, and I'll come back to that point again later, but we can train monkeys to reason from probabilistic symbols. And so here a monkey's been trained that there are 8 different kinds of symbols that come and go, and whenever he's ready with an answer, again, a reaction time experiment, he can say, I want to choose the right target, because these symbols, they confer a weight of evidence that either favors right or favors left, and the amount of weight of evidence is something we control, okay? So we assign them different degrees of reliability. It's sort of like, so the right thing to do is combine evidence, but to combine it weighted by reliability. It's kind of like, we all listen to the news, and let's say you happen to walk past some fascist, a place where they're showing Fox News. So you know it's an unreliable source, and you walk past it. But, or maybe you ask the owner of the shop to shut it off or you're not going to get your hair cut there anymore. I do that. That's my acts of civil disobedience. And, or maybe, you hear NPR or something else that you think might be a little bit more reliable, and so you might pay more attention to that. Okay, I hope I didn't offend anyone with that. I only hope I didn't offend you, not out of compassion or something, but just because I hope that you wouldn't be offended by that. Anyway, so what we do now is we study the same kinds of neurons. We know where their response field is. And I'm going to show you an example of a neuron that's accumulating evidence in this task. And this is a neuron that prefers this target. It would have persistent activity before delayed eye movement to this target. Okay, so that's the trial. The monkey's eye position was in yellow. You saw that he ultimately chose this spot. Now, of course, you couldn't remember what all those shapes were, but I'm going to tell you what this graph is at the bottom now. Those are the spikes you just heard, and I assume someone will interrupt me if you can't hear these spikes anymore. And what I'm plotting here is the weight of evidence, the log likelihood ratio, in favor of this target, the one that's in the neuron's response field. And what you can see is that the first shape favored that. This yellow is neutral evidence, okay? And this is the cumulative evidence in units of log likelihood ratio for the target in the response field. So the first shape did favor that target, and there's a little elevation in response. You might see the spikes are a little closer together than they were back here. Okay, now the next shape reversed that. It was pretty strong evidence, and it actually got us to the other side of neutral here. And so the net evidence is now favoring the target that's no longer here, the one to the right. And you can see that there's sort of a little decline here. Now, you could accuse me of reading tea leaves here with these, these barcodes. But, over many trials, we can convince ourselves that that's actually a real dip in the firing rate. And then you see there's mounting evidence starting at shape 3, 4, 5, and 6 before the monkey stopped and answered. And you can see that mounting evidence is associated with a crescendo in the firing rate. And you heard that. Okay? Let's hear another trial. So this one had kind of the opposite pattern. In this case, the first few shapes were favoring the target in the response field, okay? And you hear this rise in activity, and then followed by a decline as there's meandering but mounting evidence ultimately in favor of the target outside the response field. Now you notice what's happening here is the firing rate on single trials from a random kind of random neuron in the parietal cortex, so deep in the brain, is showing you that it's doing additions and subtractions. It's keeping track of a running sum of basically positive and negative numbers. Okay, so it's treating those as increments and decrements and showing you the accumulation of its firing rate. So don't let anyone tell you that when I show you an average that looks like a ramp, that average is a bunch of individual steps on every trial. These neurons have not only persistent activity, they have graded persistent activity that can change many times over the course of a single decision. Okay. Now, I'm not showing you all the behavior from this. You can read the paper if you're interested. Because the monkeys behave as if they are accumulating evidence in units of log likelihood ratio, which they then combine and stop when they reach a critical level, and that explains their behavior. Okay. Now, so this is, we think, is a pretty general mechanism for deliberating or really making a plan, but basing it on evidence from, in this case, the world, so a perceptual decision. It's a little bit hard to think about this in terms of just integrating evidence coming out of the visual cortex. It seems to involve some kind of associative memory that links the weight of evidence to each one of those shapes. We train the monkey like crazy so they can learn them. Again, it's not a very natural task. The only natural part of the task for the monkey, I think, is that effectively this is like foraging. He's looking for information that bears on where he's going to get his reward. Okay, so now I want to introduce this circuit configuration problem, and I'm going to do it the easiest way I know how, which is to introduce one particular kind of configuration change, which is the routing problem. Okay, so imagine that we've trained this monkey to make left-right decisions from a patch of dots in the upper periphery, near the more or less on the left of the midline, but in the upper hemifield. And he would do that by saying left with this eye movement or right with this eye movement. Let's imagine this target is in the response field of a neuron. Well, on another trial, if the monkey's trained, we might put the random dots over here and ask him to choose between northeast and southwest. In this case, now he's going to use different information to choose, say, northeast. And another trial, we might put both targets in the right hemifield and have him choose between up and down. And again, this is still, kind of like a neuron that we might expect to accumulate evidence for motion. But now it has to get its evidence from a different part of the visual field. I'm gonna look back at D1 to make this explicit. This is an old 2-deoxyglucose pattern. In other words, showing the activity of this a set of rings in the visual cortex. It's an old paper by Roger Tutel from the 80s. And let's just look at on visual cortex where these things are. So the target representation is over out here in the periphery. Okay, it's in the upper field, right? It's the upper periphery. And the northeast, southwest dots are, not too far from the vertical meridian, which is over here, and might occupy this much space given its magnification factor. The up-down over here, and the left-right over here. Now, neurons in LIP, and probably elsewhere, but a single neuron in LIP that we would record has the capacity to get information directed from all three of these regions, which are centimeters apart, okay? So how does it do that? Well, we don't really know, okay? Does it have the connections to all those parts of the extrastriate cortex? Well, probably not directly. Definitely not directly from V1, but probably not directly from MT either. Okay, so that's a routing problem, and it's essentially a word that I would say it could be stated more generally. How can the same decision, plan, or proposition, imagine neurons that represent these things, find support from diverse sources of information? Our brain has to solve that problem. It's a connectivity constraint. Everything isn't connected to everything. We are not a neural network, an artificial neural network. Okay, or a Hopfield network for sure. Okay, so also let me introduce another kind of circuit configuration change, which is that it's context, the dependence on context. How can an associative memory, for example, like the associative memory of weight of evidence with those shapes, change the level of persistent activity? So now I'm kind of stating the integration problem, one that's more like LIP receives an instruction based on the content of an associative memory. Okay, so that's a very different kind of way of casting the computation of accumulation of evidence. It's more like the language of computer science of thinking about these. The LIP is effectively like a finite state machine, and the shape, one of those shapes, the Pac-Man shape or something, triggering an associative memory that turns into more of an instruction to change the firing rate by some amount delta, which is effectively the content of the working memory. Of the associative memory. So, so one could say, how does the same information lead to one behavior or another, or contingent on a second contextual cue? So this is another way that a circuit configuration might be thought of as being under the control of neural responses. So circuit configuration to me is the neurophysiologist translation of a cognitive psychology concept called control. And I'm thinking now of a very famous paper by Shallis and Norman called Controlling and Processing. So I would argue that systems neuroscience has done a reasonably good job of advancing our understanding of signal processing. In fact, when I use the word integrate and momentary evidence and things like that and direction selectivity, we are trying to understand that at a computational level under the rubric of signal processing. But when we start talking about how a signal changes the way another circuit operates, now we're talking about controlling. And routing is an example, and context-dependent change is an example. And I think the way I think about it, it's hard to understand how that works. I mean, because these changes in a circuit configuration, although they must be signaled in some way by information of spiking neurons, because we know of no other way that neurons really communicate information, but they, at the same time, their firing rate shouldn't change very much, or the circuit would contaminate the actual signal processing. So I think this process flies under the radar of our usual techniques of understanding neural computation, which is with firing rate. Okay. So, and we thought this second one, this context-dependent change in the way a circuit operates based on a contextual cue, another cue, is one that could be studied in the mouse. And so this is our first foray into that, and that's what I'm going to spend the rest of the time talking about. And I'm going to have to rush, I guess, through the first part of it. I'm kind of losing track of my own time because all I have is my watch. Okay. Anyway, here's the task. This is a simple olfactory delayed match to sample task, and it was brought to the lab by Herbert. He goes by Herbert. His Chinese name is Zheng Wu. But I'm going to call him Herbert in this talk, and he's on the job market, by the way. And Herbert trained mice to do the simplest delayed match to sample. There's only two possible odors. I'll call them A and B. Here's what they really are. So, and the way it works is the mouse gets a little sniff for half a second of sample odor A or B. He waits through a delay, which is typically a second and a half, sometimes longer in some of our experiments. And then he gets another sniff, which I'll call the test, okay? So it's a delay. The question is there a match between these? And if there is, if it's A followed by A or B followed by B, the mouse should lick to the left port for a reward. And if it's B followed by A or A followed by B, in other words, non-match, you should lick to the right. Now, that's delayed match to sample, but I want to encourage you to think about it slightly. Oh, I want to say one other thing about that, is that it's formally equivalent to the exclusive or problem. It's the same truth table if you think about it, okay? So, and that's kind of interesting for computational people because the exclusive or is one of these poster, archetype problems for the needs of nonlinear kinds of operations. And I won't say more about that, except I'm going to show you an interesting way that the mouse brain solves exclusive OR, and it's not by embedding in high dimensions. So, so here's the way I want you to think about the problem. I just want you to consider that the sample odor might play the role as a contextual cue. And under context sample equals A, then test odor A means go left, and test odor B means go right. But under contextual odor B, then test odor B means go left, and test odor A means go right, okay? So we could think of that as a change in a circuit configuration, and I wouldn't be telling you that if there wasn't evidence for that in what I'm about to say. So here you're seeing a mouse doing the task, and you're looking from below. There's his licking. He's licking to the right port for 2 non-matched trials, and so forth. And I would just tell you that the only hard part of training these mice to do this is a lot of people do match non-match using a go no go, but we wanted a symmetric behavioral task, and the hard part is just getting them to delay their licking, okay? Because we wanted to not see lick contaminated responses during the sample and delay. Okay, so very quickly, because this turns out to be the least interesting part, which I hate to say because I am a neurophysiologist and love the sounds of neurons, but we did a survey of the neurons in just a selected set of areas that represent the odorants, some association area, and the motor response. Okay, and so ALM stands for the anterolateral motor area. It's part of area M2. Studied by Carl Swoboda especially. It contains essentially you can think of it as a frontal eye field for licking and maybe whisking too. Okay, and then and the piriform, of course, is a primary olfactory cortical area, and the orbitofrontal cortex is a classic association area. Okay, not surprisingly, in the piriform cortex, we see lots and lots of sensory neurons. Surprisingly, a lot that are just sensitive, selective for these 2 odors. And here is a neuron that responds at the time of sample, which I'm delineating with these faint dashed lines. It'll be the delay. I'm going to show you a lot of graphs like this. And then between the test is also delineated by these 2 faint dashed lines. Sample, delay, test. And so this is a neuron that responds to odor B. These 2 red, B is preceded by B, or B preceding A, I mean. And then also it responds to B when it appears in the test, when it occurs in the test. So here's, AB and BB, these 2, this pink and blue. Okay? And so, maybe there's a slight difference in the way this neuron responds to B, depending on whether it was preceded by A or B, but that's that. So is it what you might expect mostly from a sensory neuron? Many of these neurons have persistent activity that lasts through the delay. That's important because in order to solve the delayed match to sample task, you need some kind of representation of the sample at the time of the delay, at the time of the test, in order to make the comparison. Okay, well, in piriform cortex, we see a lot of neurons that are selective. And what I'm showing you here is a selectivity index based on an ROC measure. In other words, it's just sort of getting the overlap, and so if it's at 1, there's no overlap between the response to A over B, or here B over A, and so that's not surprising. We see neurons like that in the orbitofrontal cortex too, and although some of them have more mixed selectivity in the latter part, and in ALM we see very few neurons that are selected for the odor, and it's very weak. At that. Okay. So when we do see them. Okay, of course, the choice itself, which is effectively which way is the monkey going to lick, that is the dominant neural type we see in ALM. So, almost all the neurons that are modulated by the task at all are sensitive to lick left or right. They don't do much. They don't do much before that. And let's see. What do I want to tell you about that? Yeah, that's about it. So this happens to be a non-matched neuron. We see as many matched as non-matched neurons, in other words, right and left licking in each hemisphere. Okay, so here's the ALM selectivity index for licking left and licking right, or match non-match, and we see a little bit of that in the OFC as well, but it's not very strong, and very, very little of that in piriform cortex. Okay, so that's not a surprise. I want to just emphasize one thing, is that at the end of the delay, so this column here, where you really must have some kind of representation of the odor in order to solve this task, we don't really see much of that at all in ALM. So, you know, a few that make significance, but with very, very weak selectivity. Okay. Now, what is all the rage in neuroscience these days, sadly, in my view, is to train classifiers to decode information. We are recording from, you know, up to 80 to 100 neurons, often from multi-probe electrodes in these areas, and so we train simple support vector machine declassifiers because, you know, everyone wants us to do it. If I had my way, we would never do it. But in any case, we do it, and you can ask me why I feel so strongly about that later. But I'm about to make the point. But we see that these declassifiers can decode from piriform and OFC almost as well, or in the case of OFC, about as well as the mouse performs the task. By the way, every now and then I might slip monkey for mouse. I'm just so used to it, and I apologize. If I could see your faces, I would see someone laugh, but I can't do that. So anyway, so basically, we have a representation in the sensory cortex, and that's probably, you know, that little trial type selectivity, those little modulations at the time of test based on what the sample odor was, is what, you know, the decoder exploits in order to solve this. And of course, we can solve the match non-match in ALM even with single neurons, let alone with a population of them on single trials. Some of them are, you know, they're so selective, but that's trivial because, of course, those are neurons that are telling us which way the mouse is going to lick. Okay. Now, so this supports what I would call the standard hypothesis in neuroscience, which is that upstream sensory areas and sensory and association areas resolve the decision, match versus non-match, and instruct ALM. Or maybe even ALM could be the decoder, could be acting like what our SVM is doing to the inputs from the upstream areas. I'm channeling what I think is characterized mainstream computational systems neuroscience. Okay, that's not my own prejudice. Okay, so we decided that rather than just accepting that at face value, we would test it. And Herbert came up with kind of a clever way to do this, which is he uses these mice that express channelrhodopsin only in VGAT, or he kind of created these mice out of ones that express yellow fluorescence protein. But anyway, we express channelrhodopsin in the VGAT positive, in other words, the GABAergic neurons. We do that in both hemispheres when we inactivate bilaterally with the light, but only in the sample and delay period. So our logic was we ramp this down. You have to ramp down until you get some bounce back of the neural response. So we tapered this off so that by the time the test comes, which is the first time that the mouse can make the non-match decision, the cortex is back to normal. The monkey can, the mouse can lick, caught myself. The mouse can lick normally and so forth. And I'll show you a control experiment that demonstrates that. We have several, okay? So the idea here is that if upstream areas could solve the task, they can only solve the task when the test comes. And at the time of test, they have access to ALM. So we're doing this in activation. I'll tell you about OFC just as kind of a control in a moment. But our main emphasis is on ALM, okay? And here's what we found. We found that manipulation, that intervention, impaired performance dramatically. So here's the control trials, and here's the laser trials. And each line is a single experiment. Okay, so there's 14 individual experiments. This dark one is the average. And what it's showing is just the proportion correct. And you can see we went from, on average, 97% correct down to, something below 70% correct. It's a pretty striking deficit. Okay? And as I said in OFC, this surprised a lot of people. We saw much less of a deficit. It's subtle but not significant. Okay. But of course, you keep in mind that there are many areas that connect the olfactory epithelium, or the bulb, let's say, to ALM. So there are many paths to get the odor information, which obviously is necessary to solve the task, into ALM. Okay. So I'm not going to tell you about all the control experiments, but this one is, I think, the most informative, or among the most. What we do here is we train the mouse on another kind of trial where we give him two new odors, C and D. And these trials always work that C is always in the sample position, but then in the test position, C means go left and D means lick right. Okay? I should lick left and lick right. So in this case, this Q, the sample odor, or what I'm going to encourage you to think of as a context, is irrelevant. It's only the test odor is needed to solve the task. Okay, and so we interleave those trials in single experiments. And so what we do is here we're showing that we reproduce the result I showed you in the previous experiment. That is a pretty substantial impairment in performance when we inactivate an ALM on the AB task. But when we inactivate in the C crossed with AB task, we see very little impairment. A little bit in these two mice, but for the most part, not much. So let me tell you what I think we learned from these inactivation experiments is that ALM is playing an important role in this delayed match to sample task, specifically under conditions when the delay is instructive. Okay, so not, for example, in the CD task. Inactivation does not impair the animal's capacity to translate a decision about odor to a lick response. Okay, so you saw he was able to perform the task when we did the inactivation. And he was able to perform the CCD control task as well. Also, he has no impairment in licking. We can produce an impairment in licking if we keep the light on through the licking period. We can shut down licking. Okay, so then this is, I think, another important point is that the inactivation of ALM does not affect the neural responses in OFC and piriform. I didn't show you this, but if we do that same SVM decoder, we can do that even on trials where we have inactivated in ALM. Okay, so what we learned from this is that signals upstream in upstream areas, they can impact ALM. Okay, that's what must happen in CCD control, for example. But they don't resolve the match versus the non-match, despite our ability as statisticians running a machine learning trick, SVM decoding, despite the fact that we can decode. So I would pose this as a cautionary tale, because it is the rage, or au fait, whatever the right expression is, for everyone now when they have big data sets, and we're getting them in the monkey now. We're among the first users of the new neuropixel, macaque neuropixel probes. It's everyone wants to decode, information. My view is that it's basically a waste of time, because decoding is no guarantee that any downstream structure achieves this. In this case, not only does ALM not decode what the information in OFC and piriform, but we could say that any area that had access to ALM and didn't and was not blocked was also unable to send its signals to ALM to allow ALM to perform the task. So I think it's a good cautionary tale the next time you hear someone tell you that they've decoded information and they think that therefore the animal used that, a strategy like that. Okay. So I'm not arguing that you can't decode information and say that therefore the information is present in a population. I mean, it's a useful statistical tool for that, but that's a different story. So this hypothesis that I mentioned, what I would call the standard view, is that upstream areas resolve match versus non-match, and they instruct ALM, or ALM being the decoder. I think we can exclude that. And so we now consider an alternative, is that ALM is capable of implementing one of two sensory response mappings, and ALM and this computation must occur in ALM. And that a decision, and this sensory response mappings, you could think of it as a decision between two rules that test odors A and B, instruct left and right respectively, So that's under actually context A, when the sample odor is A. And or the other rule is that test odors A and B instruct right and left respectively. And that, in other words, that's when the sample odor is B. Okay, so that's and so it's the sample odor that determines which rule is going to be instructed. Okay. Now, there's a problem with this alternative hypothesis, and that is when we did our survey of the neurons, we did not really see much of a representation of a sample odor in ALM, especially during the delay, and definitely not at the end of the delay. What we saw was extremely weak. So we were worried about this, and we decided to take a closer look because Herbert had the wherewithal to realize that much of our samples were from deeper layers 2, 3, and then the deeper layers of the cortex. And so we didn't really sample much in superficial layers 2, 3. So what I'm showing you now is 2-photon imaging. This is not the raw fluorescence signal. This is a direction index computed from the calcium signal. Okay, and again, this is the sample period, the delay period, this gap between these double dash lines, and this is the test period. And what I'm showing you, the heat map shows the selectivity, an absolute value, so we don't care whether it's selective for A or B, but just the selectivity. And you can see that there is a sample in 40-something neurons in this field of about 220 neurons that 200 plus neurons, I say, that there's of the sample odor, in this case often spanning the entire delay period. Now it turns out what I'm showing you is that this is just one field, and it so happens this field was at a depth of 100 to 200 microns that we see these neurons. When we go deeper than that, we rarely encounter them. Maybe a few, in deeper layer in layer 3. Some are deeper in layer 2, 3, I should say. I'm going to refer to this because this is the first 100 microns of the first really cellular level neural level layer of layer 2, which is so I'm going to call these layer 2 neurons. This concentration of neurons that had never been seen before. We kind of felt that something like this had to be in ALM, and there it is. And it's sitting up there in these superficial layers. Now, by the way, we are sampling these other layers. I'm not showing you this, but we see Lick cells throughout all the layers. In fact, they're the dominant cell type even in this first 100 microns, okay? Now, they come in flavors. Now I am showing you the raw signal. So these are now single neurons, okay? And some of them have pretty stereotyped response. This one, responds, it's a B-selective neuron. This is an A-selective neuron, pretty stereotypical in its response. They're not perfect. There's some response to the other odor. There's some response to the other odor, right? And some of them are more variable. They respond at sort of at any time during the delay, during the sample or delay. So they kind of can span. I just ordered them in terms of, or Herbert ordered them in terms of of roughly when their peak response was, okay? And I'll mention these again, but they come in all kinds of flavors of scattering in time and duration. So this is our, a long duration neuron, a scattering neuron, and something that has a little in between, okay? Now we don't know that these layer 2 neurons are mediating the behavior. We're working on that right now because when we inactivated all the pyramidal cells. We assume they're among the pyramidal cells because we're only expressing the calcium indicator, GCAMP6F, in pyramidal cells. But this is the evidence we have that they are related to the task. In monkey neuroscience, we call this a choice probability analysis, looking at single trial activity and its relation to choice. And what I'm showing you here, for example, is intuitively, like if let's imagine that we have a neuron that prefers sample odor A, okay, and then we just rank its responses to sample odor B, which are, he's going to have some responses. When that response is on the higher end, okay, that's when the mouse makes more errors, okay? when we take the cells, the neuron that prefers A, and look at and rank it, then it's again, just take the rank order of the responses. We find that it's when it was responding, it's in the lowest percentiles that we see the error rate. So they pile up in this left-hand corner. And, it's not a big effect because the overall error rate in this experiment was only 11%. So, the mouse is performing at 89% correct. But it's statistically reliable. So that's the best evidence we have that these neurons, that their variability in their calcium, whatever's giving rise to these calcium signals, presumably spikes, but we haven't actually measured that directly, is related trial to trial to the behavior of the animal, the success or failure. Okay, so what's going on? I want to share two relevant observations with you. One is that the impairment is alleviated if inactivation doesn't span the entire sample and delay. So here, in addition to inactivating during the sample and delay, there's a condition where we only inactivated in the sample plus early delay, okay, or just the late delay. And you can see that compared to control, there's a slight decrease, but it's the biggest decrease we get is when we inactivate for the entire time. Now, with Ashok, Litwin, Kumar, we trained recurrent neural networks to perform the task, and this is just a kind of a fancy way of stating something that might be obvious to you. These curves represent whether we trained the recurrence at each level. Technically, you could have done what I'm about to say in just 2 levels, but it's kind of fun to think of it this way. So imagine that only ALM, the only areas that could have recurrence and therefore persistence were prior to ALM. And then in that case, what we predict is, well, they have to kind of solve the task. And then if we simulate an inactivation experiment, we should never see an effective inactivation because we're not inactivating the area that's making the decision effectively, that's solving the problem. Effectively, this is the logic behind Herbert's inactivation in sample through delay, but leaving ALM intact at the time of the test when the delayed match to sample decision can be made. Now, if we put recurrence only in ALM, okay, then that makes another obvious prediction, which is that with a recurrent neural network, if you inactivate at any time during the sample and delay, you should get almost complete inactivation, okay, because a complete impairment, because there is no representation of the sample at the time of test, which is what you need. With recurrent network needs that in order to be able to solve the problem. So here's what we get if we allow for recurrence in all 3 areas and train the network. And what we see is that we get effects of increasing impairment as we inactivate more and more of the delay, and especially late in the delay. But that's a problem, because that neural network solution like that, with persistent activity effectively providing the linkage between sample and the ability to compare the test to the sample, implies that in late delay, if it really knocked out all of that recurrence, then we should have gotten the same impairment here as here, but we didn't see that. So we don't really think the neural network solution is the recurrent neural network is the way to think about this. And instead, we're toying with and testing now other models. But I'll come back to that in a second by saying the second relevant observation, which I kind of already introduced, which is that the mice don't generalize to sample match to sample. If we try them with new odors like CD crossed with CD, they have to be trained all over to learn delayed match to sample. Okay, so they just learn another 2x2 contingency table, like the exclusive OR table, effectively. But if we train them on this task, which is sample odor is either C or D, and the test odor is still A or B, now we really can't call this delayed match to sample, but really it is a context-dependent change in a rule. And mice can learn that just as easily as they learn the AB crossed with AB. And this is just an indicator to us that maybe this is really the way the mouse is solving the problem. They really are treating the sample odor as a contextual cue. So with those just really an intuition, it means that something must be going on in ALM during the delay period, we think, that those layer 2 neurons are doing something to the neurons that are lick left and lick right neurons that allow it to interpret the test odor as an instruction to lick left or right. And one way to achieve this that we've played around with is with synaptic facilitation and depression. It takes more than that, of course, but that's a flavor of models. Another way to achieve this is with gating models. My favorite model is actually, this shows us like a presynaptic gating, but my favorite is postsynaptic on dendritic branches, where one could say that imagine that the sample selected neurons, the layer 2 neurons, make synapses, say, onto dendritic branches where test odor A, the information that conveys information about the test odor, A or B, would say concentrate. And then by turning on or off dendrites, in this case shown as a presynaptic control, but by turning on or off inputs to the neuron, we might to the match non-match, that is lick left, lick right neurons, we could change which inputs were going to drive the response. Okay, so these are things that we're studying now. We have no direct evidence for that, but the idea that the circuit would establish a change in the way it operates on the basis of a contextual cue is what I was introduced this work as being in the service of, and we think we now have a system for actually studying it. So let me close by saying that premotor area ALM is involved in a key step of an olfactory decision. Now, I would say that's not surprising from the work on perceptual decision making in primates, because the key insight that I get from all this work, not just my own, is that the way that knowledge exists in the brain is in persistent activity. That's how the only way it could transpire across time, and those persistent states don't live on structures that keep up with a changing world, sensory areas. They look more like propositional things about what we're going to do to the world. I call those affordances, to steal a term or borrow a term from Gibson. And I call them provisional affordances because it's not like you have to act in order to know. You just have to organize information as if it's in the service of a potential action. So the idea of finding that knowledge states or the use of information to do a behavior would live closer to the motor system than to the sensory system, I think is just a page out of the playbook from systems neuroscience of decision making. Now, upstream sensory and association areas must supply information about odor identity by the end of the delay, but they don't supply ALM with the decision. I think I've said that enough already. It's a cautionary tale about decoding. The working hypothesis now is that ALM receives input about sample odor identity to alter the effective connectivity of lick left and right neurons, such that one or the other odor input at test leads to excitation. Okay? And the layer 2 neurons are a promising candidate to mediate this change in the circuit configuration. They're the only game in town. I've shown you that they have a trial-by-trial association with success and failure, but beyond that, we don't really know that they are causal, but we're working on that now. And I will just end also saying that this somewhat complicated exclusive OR problem is structured as a decision about a decision. It's implemented as an instructed change in a circuit configuration, although we don't know how. I would say that this is a model of what's called executive function or cognitive control, and it's a control operation as opposed to simple processing. I shouldn't call it simple. We need both. And so with that, I'll stop. I'll thank my collaborators. Herbert Wu, as I said, is on the job market. Ashok Litwin Kumar did the computations and also was intellectually involved in all stages. And Richard Axel, who is an expert on olfaction and olfactory systems neuroscience, and an absolute delight to work with. My support, lots of guidance from very helpful people, especially Karol Swoboda, And you might have noticed I started and I'll end with this rare painting by Magritte. Here he is rendering his own decision process. Shall I paint ceci n'est pas un pipe or le clairvoyance? And here was his decision outcome. It's le clairvoyance. And with that, I'll take your questions if we have time.
Thank you, Mike. And if everybody wants to unmute and just join me in thanking
Mike, can you hear me? This is Dieter Jäger from Emory.
Hey.
Yeah, so we work on ALM quite a bit lately, and I have read a lot about the Swoboda papers, and now I'm getting maybe more confused than before, because he's describing this intricate feedback during delays for tactile and auditory cues between layer 6 and VM thalamus. That doesn't seem to be on your radar. So I don't quite see whether odor is just different or whether the task is slightly different, or why would he see?
Yeah, the task is different. Remember, the mouse does not know. It's a great question, and we're very aware of this work. The mouse, in all of those delayed responses, the mouse knows that he's going to lick left or right. So these are what we call instructed delay paradigms. Now remember, in delayed match to sample, the mouse does not know which way he's going to lick. If he knows anything at the time the sample odor comes and through the delay, all he knows is what rule to apply, which sensory response mapping to apply. Test odor A or B, lick left or right, or test odor A or B, lick right or left. So he doesn't, that activity is, and you could say, represented by these layer 2 neurons. They are representing the odor, but from the point of view of a motor circuit, they're representing what rule to apply. And we think what they're doing is they're readying the circuit to apply that rule. So that's the difference between these paradigms. If the mouses can plan lick left, then you see this persistent activity in favor of lick left. And Carl believes that this is supported by that loop that you mentioned. I think there are interactions between the hemispheres too. But I don't remember that story. Or maybe there aren't. I forget that. Yeah.
No, there are. And just as a little comment, because I collaborate a little bit with Matthew Larkham in Berlin, and he has a couple of papers out in 2020 about decision-making processes and context dependence. And he would certainly claim that it is dendritic nonlinearities that can provide the context on decision.
Absolutely. Matthew and I have been talking about this for years. And so absolutely, I look at the mechanisms that interest me. This is why I said my favorite ideas about them are post-dendritic, and they involve things like, calcium plateau potentials and NMDA spikes and all kinds of things like that. And I believe that this problem, this circuit, these kinds of operations, I'm going to speculate really irresponsibly now, but Matthew would agree with me, I'll bet, that this problem, the circuit control problem, is the key to understanding tons of things that we blithely think we have any understanding about in systems neuroscience. For example, feedback. There's not one single good idea about feedback in systems neuroscience because they all make believe that neurons receiving input from feeding back structures can somehow process that information without ever revealing it in their spike discharge. And they make believe that neurons that are providing this feedback can somehow pick up, address the proper neurons that they're supposed to be feeding back to. There's some kind of a handshake that has to occur between areas, and it probably occurs by neurons identifying themselves by virtue of some kind of change in the state of their apical dendrites. And the reason it has to be on apical dendrites and maybe distal basal dendrites is because, to make a joke out of it, if I was on the Celestial Design Committee, these controlling operations should not contaminate the actual signal processing. They shouldn't lead to changes in spike rate. They should lead to changes in the way the circuit can then lead to change its spike rate in the process of computing, which is the processing part. I wasn't on that design committee. I think a little bit leaks in, and I think that's what the field of attention studies with their half a spike per second modulation in V1 or something.
Thanks a lot.
Okay, we probably have time for two more quick questions from Mark Wheeler and then Garrett Stanley, and that's probably all the time we have. Mark, do you want to ask your question?
Yeah, sure. Mike, I was just wondering if you could say a little bit more about the Alan Turing work, the Enigma.
Oh, okay. Very quickly, the process itself is the accumulation of in units of log likelihood ratio or products of likelihood ratio to a threshold. It's called the sequential probability ratio test. It's the most efficient test for deciding between two hypotheses if you can do it, and with the least number of samples, efficient in the sense of given a desired level of accuracy. Turing, in the one step of the decoding of the Enigma machine, was to find random letters that were intercepted, okay, that would have been produced by Enigma machines in exactly the same state. And it turns out that if you line up letters one above the other from these random things that haven't been decrypted, we don't know what they say, then the probability of seeing the same letter, even in the mutated or code, the encrypted versions of original messages, will, by this deterministic operation that the Enigma machine worked in, they'll preserve probability of matches. So he accumulated evidence from pairs of letters that matched versus non-matched under the two hypotheses, Enigma machines in the same state or in effectively random states. And he accumulated in log likelihood ratio until some threshold, because they just had to get through as many as possible. And by finding two Enigma machines in the same state, from that they could figure out what the code was for setting the rotors, and from that they could decrypt all the messages from the day. And if you watch that horrible movie about Turing, and you see these women on these flat tables sliding things around, they were basically sliding around pairs of letters to see and finding out whether they could provide support or refutation of the hypothesis, machines in identical versus random states. He wrote that in the back of a piece of paper, but his deputy statistician, who became a very famous Bayesian, I.J. Good, he went by Jack Good, he wrote about that in a book called Good Thinking that I ran into this by accident.
All right, thanks.
All right, great.
Yeah.
Thank you.
Oh, yeah. So, hi, Mike. Garrett here from Georgia Tech. So mine was more of a comment than a question. I was just, as you were talking about the fact that the olfactory task that you've designed here couldn't really generalize for different odors, I started thinking about the fact that, if you had chosen a different pathway, you might be able to titrate this so you could play around with the quote distances between the sample and the match. And I was just curious, I guess olfaction, you're thinking it's more about identity of odor rather than something along a continuum that you could measure and play with in terms of the distances between these two. Just maybe more of a comment.
Yeah, no, it's a great comment. It's not this is something that one might want to do, try to understand distance and odor space or chemical space and so forth. They're kind of interesting questions. It's the kind of thing that I absolutely wanted to avoid. Remember, I came to this looking for a context-dependent change in a circuit configuration. I wanted to study that because I see it happening every day in a monkey, and I gave you kind of a simple example of it. But I wanted to understand it because I think I have to do cell biology and really the fine-scaled kinds of manipulations that are not available to us in the monkey, so I thought I could do it in the mouse. So we had our mice actually achieve the task, and a lot of us, some of us in the project, were hoping that we could solve the task with the SVM decoder stages and things that were going on in piriform. But I would always remind them that the moment this problem was solved upstream of ALM, which was my favorite hypothesis from the start, you see, or some area, parietal area, influence to ALM or something, but that I would just abandon the project because I wasn't interested in studying distance and similarity in the actual perceptual side. I can do that in a monkey and way better than in a mouse. So I'm not saying people shouldn't do this in a mouse, by the way, but I'm just saying that wasn't what I wanted to do. I want to study this problem I can't study in a monkey, which is the problem of effectively change, a control problem.
Yeah, got it. Thanks. That helps clarify sort of the motivation of the choice.
And by the way, we can train them, and we haven't tried hard enough. I think the furthest out we went was 3 different odor pairs So if if we got out to 10 odor pairs for a mouse, and assuming he lived that long to get it all, then maybe the mouse could generalize. But that's not the mouse I want to study.
Thanks.
All right, thank you so much. So, Mike, you have a couple meetings lined up, and the first one is with graduate students that starts, well, it was supposed to start 4 minutes ago, and I think Terry probably sent other links that are Zoom links so that people can actually see you. But again, thank you so much for accepting our invitation and for giving this wonderful talk.